Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about neurons, nerve cells, and then we will, in our next talk, get into the nervous system and how it all kind of gets put together. So let's take a look at just kind of a nervous system overview. Nervous systems and nerve cells are only found in animals, and the reason why they are in animals is because animals need to communicate quickly over long distances. You think about our bodies, right? Our bodies are made up of trillions of cells, and we're between a meter and 2 meters from the ends of our bodies. We want to be able to communicate over those long distances, and we need to do so quickly, especially because we have our brains that are controlling everything, and then we've got muscles like in our legs and our feet that we want to be able to move. That movement needs to happen pretty quickly. Things like the hormonal system, while it's good for long-term responses and for regulating a lot of things, it's not good for those quick changes, quick responses to our environment. And so we've got our nervous system. It's fast, it's very accurate, and it has a very quick reset. And we're going to take a look at all of those different things. But let's start off with just the basic neuron, right? The neuron is the baseline cell of the nervous system. It has kind of some main features. It's got dendrites, which are these kind of the inputs of the neuron where it receives signals. You got the cell body, which has all of the things that you come to expect. You've got your nucleus, your organelles, basically all your normal cell stuff. And then you've got the axon. The axon hillock is where the axon attaches to the cell body, and the axon is what transmits the signal to the next neuron, where it will either attach to the dendrites or directly to the cell body. There are a couple different types of neurons. We've got sensory neurons, which have pretty extensive dendrites, but they have a much longer axon, and the cell body is kind of in a spot along the axon. Sensory neurons are neurons that are going to be things that are connected to like your eyeballs or to your nose, things like that. Then you've got the interneurons. The interneurons are found in your brain. They have lots of dendrites, a very extensive dendrite system, and then a smaller axon comparatively. And then finally you have your motor neurons, which are found in things like your muscles. Just some random facts about neurons. They are the most highly specialized cells in animals. They are also the longest cell. There are some blue whale neurons that can make it up to 30 meters. Giraffe neurons can be up to 5 meters, and you have some human neurons that are 1 to 2 meters long. This right here is a color graph showing you some neurons. So these would be dendrites up here. Here would be some cell bodies, and these are axons. And this would be found in the cortical portion of the brain. So transmitting a signal requires two different things. We've got resting potential and action potential. And neurons do send electrical impulses, although the way that their electrical impulses are created is through chemical means. And I'll explain that as we move forward. But during resting potential, no signal is being passed. This is just kind of the stationary. The ion pumps and channels are used to establish that resting potential, and the resting potential is between -60 and -80 millivolts. The action potential, this is where the signal is actually being passed along. What happens is that the gated ion channels are going to open, and it has a voltage of around 40 millivolts. And so you can see the difference between the resting and the action potential, and that change is what is going to actually allow us to pass a signal. So resting potential, you've got anions and the potassium being more highly concentrated inside the cell, the sodium and the chlorine being more concentrated outside the cell. And what that does is it polarizes the membrane, so you have a positive charge outside the cell and a negative charge inside the cell, and we have some potential energy that is stored up. And that potential energy, think about it being kind of like a waterfall, right? We've got a dam in the middle. We've got more highly concentrated amounts of either of these substances on both sides. And so when we open up the dam, that allows us to transfer energy, which we can then use to submit a signal. Now, how does the actual resting potential become formed? Well, if you look more closely at the membrane, what you find is that there are more potassium channels than there are sodium channels. All right. So what that means is you have a net movement of potassium ions from inside the cell to outside the cell. Now, the potassium-sodium pump helps to maintain a concentration gradient by constantly pumping those potassium ions back across, while also pumping some of the sodium ions back out of the cell as well. And that is able to help us maintain our concentration gradient, where we have high amounts of sodium inside the cell, lower amounts outside the cell. Now, the way that the net movement of the potassium ions builds up the charge is that the positive charge of the potassium moves out, but it leaves behind a negative charge, right? So we've got positive charge flowing out of the cell that leaves behind a negative charge inside the cell. And so overall, we get the negative charge built up on the inner membrane. Way back when, if you will travel back with me to 2.4, I talked about the sodium-potassium pump. Another really classic example of a protein pump is the potassium-sodium pump. This is definitely one that you want to be aware of how it works. Basically, what happens is that in your nerve cells, you have a high concentration of sodium or potassium inside the cell and a high concentration of sodium outside the cell. And in order to drive the action potential of a nerve, you need to maintain that. Concentration. So what happens is that we have a protein that fills up with sodium. Then we have an ATP that comes along. This phosphorylation word, that's just where the phosphate group from the ATP breaks off, and it forms a phosphate and an ADP. The phosphate then, once it is bound with the protein, causes a conformational change or a structural change that then releases the sodium outside the cell. When it does that, the protein has changed shapes so that now the potassium that is outside the cell can bind. The phosphate group then gets kicked off. The potassium is then released back inside the cell, and the cycle can continue. So you see that all the things that we've talked about kind of build on each other and eventually come back together. So signaling steps. So here is where we're actually going to now beginning to send a signal, and we're going to get into how action potential works. So you've got a stimulus in the dendrites or in the cell body. That signal is then passed through the cell body and sent to the axon, and that axon is then going to initiate an action potential at the axon hillock. The axons transmit the signal down the length to the synapse. The synapse is the space between one axon and the next cell, and then the synapse is going to relay the message via neurotransmitters. We're going to talk about each of those steps. So the action potential, again, it starts at the axon hillock. It is electrical, although it does involve the ion exchange. It is a binary signal, so it either is on or off. There's no in between. It's either completely on or completely off. And you can kind of think of it being like dominoes. So what happens is that when the signal first starts, it's like the first domino is falling over. And then as the signal propagates down the hillock, it's like the dominoes are knocking each other over. But just like in dominoes, it's not that the dominoes are necessarily moving from one place to another. Rather, they are transmitting their energy in a waveform down the line. And it's very similar to what is going on as an axon is transmitting its signal. And then we reset, so the dominoes have to be set up again before the next signal can be passed along. And the nerve system each neuron has a built-in reset system. So in order for the stimulus to actually reach action potential, it has to cross the threshold. The threshold is around negative 53 millivolts. And so we can have a couple of different things that can happen. We can either have some hyperpolarizations. Here's where we get an excess movement of potassium across the membrane that's going to build up more negative charge. We can have some depolarizations where you may have a little bit of signal, but it's not enough to really push us up to the threshold. Or we can have a strong depolarizing stimulus that is actually going to get us up to that threshold level that is then going to initiate the action potential, which is going to propagate the signal. The depolarizations, they will send a signal a little ways, but they don't send it far enough for it to reach the synapse and then be able to trigger the next neuron to actually act. So here's how this is going to work. We're at our resting potential. So we've got everything is where we were. Our gated channels are closed. We've got some diffusion across that's building up our resting potential. We have a stimulus. That stimulus is going to depolarize the membrane. It allows some of the sodium ions to move through the membrane, okay? As some of the sodium ions move across the membrane, they're going to be transferring charge across to the other side. And that is going to act in the opposite direction, is going to give us a positive potential. And if that stimulus is strong enough, we will reach the threshold. Once we get to the threshold, all of the sodium gates open almost instantaneously, and all of the sodium that was at a high concentration outside the cell is going to flood inside the cell and is going to flip the polarity of the cell. Because now you've got all the positive charge moving across the cell membrane in the opposite direction. The negative charge builds up on the outside of the membrane. The positive charge is now on the inside of the membrane, and that is our action potential happening. And that is called the rising phase. In the falling phase of action potential, the sodium gates get plugged up, so no longer can sodium move through the open gates. The gates are also beginning to close at this time, and the potassium gates are going to open. As the potassium gates open, now potassium is flowing out of the cell, and that is going to flip the polarity again back to the resting phase, back to the resting potential. And then we've got what is known as undershoot. So here the potassium gates stay open for a little bit longer. It kind of brings us back down below the resting potential, and then equilibrium in terms of resting potential, not actual equilibrium, but we slowly equilibrate back up to where we were when we started. And so that is the full cycle, and this happens every single time that a stimulus is sent along, and this takes about 5 milliseconds. So the total thing takes about 5 milliseconds from one place to the other, to go from one side, from step one back to step one again. So how this is sent along is that the initial nerve impulse, this is the action potential, it changes, the change in charge opens up the next sodium gate. So basically you've got one sodium gate opens up, and then that causes the next one, and the next one, and the next one. And so that wave moves down the neuron, right? Then we have the reset where the sodium needs to move back out, the potassium moves in. As the potassium moves out, that is able to then flip the charge back to where it was, and we use the sodium potassium pump in order to move the ions against the gradient. Because what happens is that as these ions move, they're not completely equilibrizing, they are just moving for a short period of time. So then we have to move them back against the gradient in order to get back to our reset point, back to our resting potential. So the potassium channels open a little more slowly. The potassium ions diffuse out of the cell, the charges revert, and then the movement of the potassium stops the sodium from moving in the wrong direction. It keeps the sodium back on the outside of the cell and stops it from coming inside. Now, if you were to just have a nerve by itself, it would not be able to do the best it can. So what we have these myelin sheaths, which are produced by Schwann cells, and those insulate and help speed up the transmission of the signal. And the way that it kind of does that is through these nodes of Ranvier. So what happens is that you get action potential not occurring at every single point along the cell, because think about it, right? We said that that takes 5 milliseconds. So if that's happening at every single point along the cell, along the axon, then that's not going to be able to be very fast, right? Instead, what we do is we have these insulated parts of the myelin sheath that transport the signal from one node of Ranvier, which are the sections where the myelin sheaths aren't found, to the next. And so it kind of hops down the axon from one to the next without having to travel along every single little piece along the way. And what this does is it allows it to be more efficient and more speedy. If you were to take a look at myelinated versus unmyelinated axons, myelinated axons move at about 330 miles per hour, whereas your unmyelinated axons move at about 11 miles per hour. So that's a pretty big difference. And the nodes of Ranvier are only found in the peripheral nervous system. They are not found in the central nervous system, which we'll talk a little bit more about when we get into the nervous system next. All right, so we've got our signal. It's been sent down the axon. Now it's at the synapse. The synapse is a little gap. It is only about 5 nanometers across from one nerve cell to the next. And what happens is that the action potential is going to depolarize the membrane. When it depolarizes the membrane, it is going to open up some calcium channels. And those calcium channels are going to allow calcium ions that are in the interstitial fluid to move into the cell. And this is part of the reason why calcium levels are very important and why it's very important that you have enough calcium in your diet, because you need it in order for proper nerve function. We talked about this when we did the little activity the other day. So the calcium channels are going to promote the neurotransmitter vesicles from fusing with the membrane. So these neurotransmitter vesicles are going to fuse with the membrane. When they fuse with the membrane, they're going to release the neurotransmitters into the synapse. Those neurotransmitters are then going to bind with protein receptors, which are found on the next cell. Those are going to open up an ion channel, which is going to allow sodium and potassium ions to flood in opposite directions of each other. And that is going to then propagate the action potential next down the road. And then the neurotransmitter is either degraded or absorbed into the cells. Now, there are a lot of different neurotransmitters out there. There are some major ones, and we're just going to take a look at 3 of them real quick. We've got acetylcholine. It is used in skeletal muscles as well as memory formation and learning. You've got dopamine, which affects your sleep, mood, attention, and learning. It has been linked with Parkinson's and schizophrenia. And then you've got serotonin, which also affects your sleep, mood, attention, and learning. And there are lots of different types of neurotransmitters that can be used. They can be affected by how strong the signal is, whether or not there are certain amounts of inputs of the signal, all kinds of different things. Now, there are some problems with neurotransmitters, and these are the weak points of your nervous system. So the neurotransmitters can be mimicked or inhibited by lots of different substances. So, for example, you've got amphetamines, caffeines, and nicotines. These are all stimulants. So they stimulate your nerve system. You've got quaaludes, alcohol, and barbiturates, which suppress your nervous system. You've got hallucinogenic neurotransmitters such as LSD or peyote, which are going to mimic neurotransmitters and cause your brain to be firing when maybe it shouldn't. You've also got SSRIs. These are selective serotonin reuptake inhibitors, such as Prozac, Zoloft, and Paxil. And then you've got poisons. And if you want to learn more about poisons and kind of how they affect you, you can go to this cool website, venomdoc.com, and you can see all the different places that these different venoms, whether or not it's from scorpions or from jellyfish or from snakes, spiders, whatever, how they actually affect your neurotransmitters and how they work. Finally, we've got 2 different types of impulses. We can either have excitatory or inhibitory impulses. Excitatory impulses can work together in order to reach active potential, whereas inhibitor can actually work in the opposite direction and help reduce the stimulus in order to prevent action potential from actually occurring. And this is where we get a lot of the complexity of the brain and other nerve systems in terms of promoting or stopping nerve impulses from being transmitted. So that's all I've got for you today. Thanks for watching. Next time we'll get into the nerve system, and I'll see you next time.